So here I am, dressed up to illustrate innate recognition. If you walked into a bank and found somebody looking like me, you would be a little bit nervous. And that's a really good introduction to the difference between how the innate part of the immune system recognizes threats and how the adaptive part of the immune system recognizes threats. If you see somebody in a bank dressed like that, you don't care who they are. You wouldn't have had to meet them before. You might never have even set foot in that bank before, and yet you would have a bad feeling. So an entire branch of your immune system works in much the same way. They have a series of what we would call pattern recognition receptors, ways of recognizing a pathogen that are common to a broad group and don't depend on recognizing any one individual. It's like good, solid, basic police work. The other one will be the adaptive, and after we look at the innate system for a bit, we'll look at the adaptive, and then we'll talk about how they get together.
An important general strategy for recognizing and destroying pathogens is to look at their overall patterns or properties and target them from that. We're going to just look at a few introductory examples to give you a taste of it before we continue on to looking at cells and processes in detail. Here we have neutrophils, and you can see that they have phagocytized bacteria. You can see clusters of these little reddish spots right here. Notice also that these neutrophils have very complex nuclei, and so here is an arrow pointing to a single nucleus that shows 4 different lobes. That's also characteristic of neutrophils. Neutrophils are a very important first line of defense. They patrol both the circulatory system and the tissues and target pathogens by recognizing danger on the pathogen surface. Here we have a neutrophil that's in the process of grabbing on to 2 chartreuse bacteria, and we can see we've got a scanning electron micrograph that shows a small portion of the neutrophil surface. An important part of your defenses are basically to set up secure boundaries between you and pathogens. One of our toughest problems is that we have to get food, water, and air into our bodies while keeping the pathogens out, so that many of our innate defenses are concentrated at the body's boundaries: skin, respiratory, digestive, and excretory linings. This picture shows the surface of skin with fine vellus hairs. Our skin is the largest organ of our body, and we'll look at it in detail in later lectures. But for now, I want to point out that skin is not some form of static shrink wrap. It's an active agent secreting oils, sweat, and defensive compounds. We also have to protect the mucosa of our gut and lungs, and we do that with a number of agents, but an important one is mucus. And an important element in mucus is lysozyme. Lysozyme is a hydrolytic enzyme that cuts up the cell walls of bacteria. It clips the wall component, peptidoglycan, that's found in bacteria and certainly not a part of your own biochemistry. What if a pathogen can get past these defenses? Well, we have internal defenses as well. If a pathogen gets into your bloodstream, there is complement lying in wait for it. This is kind of like a smart landmine. Bacterial surfaces trigger the assembly of pores that penetrate their plasma membranes and kill them. This structure here is called a membrane attack complex, and we will spend a whole lecture on these in the next unit. Finally, we have a number of pattern recognition molecules that are found on the surface of cells and in the cytoplasm should things go awry in there. A common element or motif in many of these molecules is the leucine hook, which we will see again when we look at specific examples of how these sensors activate cells when they sense danger. You can see that we don't just have one innate response. We have a constellation of innate responses, and they all work together. They also cooperate with the adaptive immune system. Here's a graph showing you the general timing of the responses: the innate system in yellow, the adaptive in blue, and of course green where they overlap. Because innate defenses are always in the ready, that is, we don't have to rearrange DNA to get them, We also don't have to undergo selection processes to help regulate these things. If a new pathogen arrives, then the innate response will take place pretty much immediately, as you can see here in yellow. And so, again, one of the advantages of an innate response is that it's very quick and it identifies things based on properties you sort of knew about, genetically speaking, in advance. The adaptive response, shown here in blue, includes synthesis of antibodies and T cells. It takes roughly 2 weeks to kick in, and if that were your only response, you could easily be dead by then. So the thing that keeps you alive before these things get going is your innate response, but that may not be enough. Your adaptive response may need to ride to the rescue. It is uniquely effective in that it very specifically targets particular pathogens. If we were to use a policing analogy, it's more like what we would have with fingerprinting, DNA fingerprinting, mugshots, facial recognition, text analysis, voice patterns, ballistics. Like the adaptive response, all of these policing functions are something where you have to have exposure to a particular bad guy first, and then you can mount a very specific response to a specific target. Another nice thing about the adaptive response, as we can see here, is that it retains a memory of this pathogen. You can see that the next time we get a repeat of this pathogen, again, the innate system is up and running very quickly. But in terms of strength of the attack, it's just deja vu all over again. The adaptive is also up and running quickly and produces a stronger response than it did the first time. Also, we'll see it will be a more effective response as well. So these are both issues we're going to look at in later lectures. We're going to explore the innate system in more detail. We're going to explore the adaptive system in way more detail. And I want to remind you before we get into any of this that these two systems work together using two different approaches to save you from bad guys. This table summarizes their differences. The innate is fast, the adaptive is slower, especially the first time. The innate is always there. The adaptive requires gene rearrangement followed by a selective process, and that's going to take up to 2 weeks to get up and running and effective. The innate system recognizes molecules by general properties. They respond to these molecules, and the pathogens will have them, and you won't. And the nice thing is then that they will never attack you by mistake. However, if we look at the adaptive, it recognizes very specific parts of proteins. You make proteins, I make proteins, pathogens make proteins. But the adaptive response will recognize specific shapes or parts of amino acid sequences of these proteins and will react and remember them when something foreign shows up. If, however, they get confused, you can get an autoimmune disease. So what we have here is an innate response that involves white blood cells like neutrophils, macrophages, NK cells, and also uses barriers and defensive compounds. All of these things we will look at in lectures 2 and 3. Later, we'll look at adaptive responses, B cells, production of antibodies, T cells, and the coordination of overall responses. Before we go looking into the specific cells that are involved in immunity, we're going to start out and look at how we get to them in the first place. That is, we're looking at the source of it all, hematopoiesis, shown here as a branching family tree leading to different types of cells. And here is the source of them all, the hematopoietic stem cell, or HSC. This is referred to as pluripotent because of the variety of its potential fates. But it's not what we would call totipotent because it can't make any old thing. For example, it can't make gut or brain cells. But you can see that this particular stem cell has a whole bunch of different options, but again, all of them are blood cells. By the time you're born, these cells have taken up residence in your bone marrow and are producing the variety of cells that you need to survive. These guys are really very remarkable. If I transplant as few as 50 or 100 of these cells from a bone marrow donor to a recipient, they could regenerate the entire blood cell population of an individual. But how do they know what to do? This is an information transfer that requires three major categories of proteins. Signals, and those will be produced by the cells around the hematopoietic stem cell, in particular fat cells in the bone marrow. Receptors. The cell has to have a receptor for this information signal or it won't be able to hear it, essentially. This signal and reception, once the hematopoietic stem cell receives something, it will change or activate or alter its transcription factors. These are proteins that bind to the chromatin in the nucleus and determine which messages are produced to make RNA and ultimately get translated into proteins. At this stage, the hematopoietic stem cells do not have surface proteins characteristic of differentiated cells and are called Lin- for lineage negative. They remain that way as long as they receive signals to remain undifferentiated. The signals induce them to maintain production of the transcription factor BMI1, which will hold them in the HSC state and allow them to divide and produce more of their own kind. However, signals called for the production of more blood cells of any type will cause some of these HSCs to produce the transcription factor GATA2. This allows them to begin differentiating. But into what? The first decision that this newly activated cell has to make is whether to become myeloid or lymphoid. If the cell expresses the transcription factor Ikaros, it will proceed down the path to making lymphoid cells. These cells begin to display molecules characteristic of the lymphoid lineage on their surface and are now called Lin+. And the cell itself is now a common lymphoid progenitor, or CLP. Notice that this cell can produce both NK and dendritic cells, which are innate, as well as adaptive cells, the T and the B cells. There will be many more signals and decisions to come before this group of cells reaches maturity and their specific functions. Now, what if a cell does not upregulate Ikaros? Well, then it's going to become myeloid and turn into a common myeloid progenitor. It is now, by definition, Lin+ because it has cell surface markers characteristic of myeloid cells. So both the CLPs and the CMPs are Lin+, but they're Lin+ because of very different surface molecules characteristic of their lineage. All of these myeloid cells are innate. However, if we look at the results here, we can see that the red blood cells come from this lineage. They're sometimes called erythrocytes, and they don't even have nuclei at maturity. The platelets or thrombocytes shown here are just cell fragments. What this means is that the rest of the cells, and that includes both the lymphoid cells and all the rest of the myeloid cells, are what are called white blood cells or leukocytes. That is, what these cells have in common is that they're full-blown cells with nuclei and the complete eukaryotic set of organelles, but they don't have any hemoglobin in them to make them red. So when you call something a white blood cell, all you're saying is that this is a complete cell and it doesn't have hemoglobin in it. That could be almost anything. When you get a blood test, you may get data on the relative numbers of which white cells you have in your blood, and that can tell a doctor or a technician a lot about your health. Before we explore these white blood cells and all their glorious variety, let's take a quick look at the red blood cell and the platelets. If you take a blood sample and look at it, the majority of cells you see will be red blood cells. This provides a dramatic illustration that hematopoiesis determines not only what types of cells the HSCs give rise to, but also how many of them will be ultimately produced. Red blood cells, or erythrocytes, primarily function to transport O2 or oxygen on hemoglobin. They also play a minor role in cooperating with the complement system to remove dead pathogens and debris, and we will touch on that in the appropriate lecture. Now, here are two megakaryocytes in the bone marrow. Notice that they are surrounded by different types of cells, including clusters of red blood cells. Megakaryocytes function to help in the repair of damage to the circulatory system, particularly that caused by trauma of some kind, but disease as well. Here I have a cartoon that we are using to represent the characteristics of a megakaryocyte. Notice we have portrayed it as a public works worker with a thrombopoietin or TPOR receptor on the hard hat. Thrombopoietin is the signal or cytokine that stimulates the megakaryocyte to divide, differentiate, and begin budding off platelets or thrombocytes. Here is a blood smear showing platelets, and you may not be very impressed. These are the very small packets of darkly staining cytoplasm in amongst a whole bunch of sort of beigey red blood cells. So you can barely see them on this blood smear, and I have pointed to a number of them using T9C orange arrows. These little packets of cytoplasm enclose signaling molecules that promote clotting. They break up when they encounter a rough surface, and here in our cartoon, we have a hole in the capillary. And when that happens, collagen fibrils from the basement membrane stick out, and you can see that one of the platelets has already stuck to the torn edge where it will burst. And that will trigger a clotting cascade. We're going to close with a time-lapse micrograph of megakaryocytes budding off platelets. It's pretty cool. And this is just a reminder that this is just the beginning of your cellular defenses. And next we'll look into granulocytes and then begin to cover the rest of the white blood cells that you have in your circulatory system.
Hello, and I'd like to talk to you about how to use the snipping tool to do a screen capture. There are other ways to do this, but the easiest way is the built-in screen tool. And I'm doing this on Windows 8. The snipping tool exists on earlier versions of Windows as well. And so the gestures and the UI might look a little different, but it's about the same. You find the snipping tool. So I'm going to hit the Windows icon, and I'm going to type the SN, and it brings up the snipping tool. And so here I go, and it's giving me this option to say snipping tool. And I'd like to say I'm going to do a new window snip. And now it's kind of graying out the screen and following me around with this little red line. And I click on this, and it has made a copy of that window. Now snipping tool is just the snipping tool. That window still exists, but this is now, I didn't want to do that. This is now just a drawing tool. So here you are. We could save this, but the other thing you can do, and this is in the more recent snipping tools, is you can make changes. So let's say, for example, you didn't want to show your name, and your name was in this path, and so I will just go and scribble my name out, like with this little scribbling tool. Pretty sweet, huh? So there I scribbled my name out. I just happened to have my name in it, so I didn't want to put it in. So now I'm going to do File, Save As, and of course find somewhere. I'm going to put this on my desktop, and I'm going to call it notepad.png. Now you don't have to put the suffix of .png or JPEG, but it's good if you're moving files between systems to put these suffixes on. Windows would know this was a PNG file, but .png is a way to nicely tell other systems when you're moving it around that it's a PNG file, or if you send it as an email or whatever. So I save it. I'm saving it on my desktop, and there we go. So it's hiding the .png, but I can look to make sure the PNG is really there, and so there it is. It's a .png, and so there's a file, and away we go. Okay. So there's the snipping tool. If I close this window and I click on Notepad, you will see that it's got the little scribbling thing. Thank you for that. Okay, so there you go. That's how to do a real simple snipping tool.
All organisms must defend themselves against pathogens. Whether you're a plant, a fungus, an insect, anything, just as much as fish and mice are attacked by viruses, bacteria, and other microbes, so you too are vulnerable. So all organisms have defenses against pathogens. And there are similarities in these defenses that suggest everything evolved from common sources. However, these, which are called innate, have their limitations. Now, in addition to innate immunity, vertebrates have adaptive immunity. That is an additional form of immunity that involves changes to the DNA and the production of antibodies and specialized cells. So this group includes the fish, the amphibians, the birds, the reptiles, the mammals, including humans. And I would like to point out that one of the reasons that we have such very large mammals, or reptiles and big birds, is that we have an immune system that's very effective at defending us, and so we're allowed to produce this great big huge yummy body that's very attractive to pathogens, and we can defend it against their incursions. Insects are also a group of highly successful multicellular animals. They probably have the greatest number of species on the planet and the greatest biomass, even though individually they can be quite small. They also have a form of immunity that allows for a flexible response. It doesn't use changes to the DNA, so it's not called adaptive, but what they have is splicing options for an mRNA precursor. That is, many of the insect's immune molecules are coded for by a gene called DESCAM. And DESCAM can produce a message that can be spliced in over 38,000 different ways. So they can produce a very large variety of molecules in response to a particular infection. So we have these defenses. Now that's the good news. The bad news is that they're energetically expensive. So here I have a picture of ATP. You can see the adenine base here, ribose sugar here, the phosphates extending outward. And the two high energy bonds are where these two phosphates are attached together. And this provides the most common source of energy transfer in all living systems. However, we can also use guanine where the adenine is, or uridine, and we can still use the high energy phosphate bonds for the GTP and ATP to transfer energy, just that ATP is the most commonly one used. So what I'm reminding you is that we are funding a policing function with this stuff. That is because the immune system, like any policing system, costs you, and in this case it doesn't cost you dollars, it costs you high energy phosphate bonds. So one of the downsides of our wonderful double-pronged immune system is that it's really quite expensive. Another downside is that these defenses represent a serious threat to your own body, and you control them to make sure they don't wind up attacking your own tissues. That does happen with autoimmune diseases, but how you then control your immune system is very critically important to you and your continued good health. The next thing I want to emphasize is that an ounce of prevention is worth a pound of cure, and I will be praising engineers, particularly civil engineers, because what they provide for us is clean water and proper sewage disposal. You put together clean water and proper sewage disposal and you get what we call sanitation, the ability to keep your pathogens out of your drinking water. When the British Medical Journal polled a bunch of healthcare workers in Britain in 2007 and asked them what was the greatest top medical milestone in the last 150 years, sanitation was overwhelmingly the favorite response. 90% of the health difference in third world countries versus first world countries comes from the fact that we have clean water to drink and they don't. So we want to be very, very grateful to engineers for this. Another improvement that I would like to talk about is something to make it another shun: habitation. We are designing our dwellings to make them harder for insects to get into. Now, here we have some mosquito netting. Mosquitoes are vectors for some of the world's nastiest diseases, and a major prevention of disease transfer is facilitated by giving you a safe place to rest and eat and sleep. And so this is mosquito netting. We put screens on the windows. I can save a good word for television because it keeps people inside. In general, that means that they don't get bitten by disease-carrying mosquitoes. And if one of the mosquitoes gets into a house with a sick person, it has a harder time getting back out diseases in its gut. Another important gift of engineers is our infrastructure. The delivery of food, proper nutrition, providing services, the ability to communicate, all of that is dependent on a highly sophisticated infrastructure that allows us to move goods and services, to communicate with each other, to provide food for everyone, because again, malnutrition is a major contributor to disease. And so if you'll recall, we need this kind of underlying support that we have in first world countries especially, but really anywhere, so that we can get goods and services to people who need them, and that will help to keep them healthy. My final shun is vaccination, and this is somebody getting an oral polio vaccine in India. They were having a vaccination program for polio. And while engineers didn't give us this one, without the communications and transportation infrastructure, we would have a very hard time delivering the vaccines to people who need them. And I want to say again how sad and awful disease burdens are on a society. They're a horrible drain in terms of personal misery, and they incur huge economic costs, because having your workers sick or worried about somebody who is sick, that makes for a much less productive workforce. And I think this picture says it all. It shows a mother who's clearly very worried about her child. It shows other people that are there in this hospital in Burundi trying to get treated. So that if children are sick, especially in infancy, that also seems to be correlated with a lowered IQ. In the 20th century, people observed not only a steady increase in the average height of people in first world countries, but also in their IQ test. And there were a lot of speculation as to what was going on. When people re-examined the data, they realized that this increase, called the Flynn effect, was paralleled by an increase in public health. And about 67% of the increase in cognitive abilities tested by IQ test could be correlated to the improved health care that people were receiving. In particular, the prevention of diarrheal diseases, okay, that sanitation issue again, is important in the proper development of young children. If you have diarrheal diseases, that robs the brain of critical nutrition at critical points in its development. Something like 87% of the energy a newborn takes in is used in the brain. It's still 44% at 5 years, and even you and I as adults are using something like 25% of our energy to support our neural functions. Other diseases damage the brain directly. Malaria, for example. If the parasites get into your brain, they can produce blood clots that can compromise cognitive function, cause strokes, and even kill you that way. So again, preventing disease is an important part of public health. And here is one of our greatest villains. This is a child with measles. And this is a particularly nasty disease for kids, because not only does measles make you sick and sometimes kill you, measles also compromises your ability to fight off other diseases. That is, it's an actual immune compromiser during the period of time and for some months after that you have recovered from the disease. Measles vaccination in developing countries can lower the subsequent mortality to other childhood diseases by as much as 80%. Just not being sick with measles makes it that much likelier that you can get over some other disease. There's currently a measles initiative working to eradicate the disease, and I would be happy never to see another case again. Now, this is what we really want for our children. We want them to grow up well-nourished, free of disease, ready to learn and grow. And underlying this course, you will see that one of my objectives always is to give you the information you need to support your country, your family, your children in the best way possible. In this section, we're going to do a quick survey of some of the bad guys. That is, things that would like to make you the meal, pathogens. And we're going to look at them in 5 different categories, because in some ways that's a good way to make sense about how they are structured and therefore what kind of attack you can level at them. And so in the first place we're going to start out with is one of our least favorites, that is viruses. And here we have a selection of unpleasant viruses. If you look at them, they all have in common that they have some nucleic acid in the middle, but they're not cellular, they're not alive. They have protein coats on the outside, but those protein coats are there to get into you. And when they get into you, your body has to find some way of telling them apart from you. And we're going to see one of the principal ways we do that is by looking at their information. So basically, these guys are an information packet in a delivery system, and their only, well, anybody's only purpose in life is to reproduce. So that's your viruses. They're not cellular. The next category, the bacteria, and this is a picture of a spirochete, are organisms that we call prokaryotic. That is, if we look at their structure in detail, spirochetes, for example, are the things that cause syphilis. Okay, here is a diagram of a bacterium, and you can see that it has its DNA not surrounded by a nuclear membrane. You probably can't tell this, but its ribosomes are different from yours, and that's why we have antibiotics that work, because they attack protein synthesis in prokaryotes and not eukaryotes. They have flagella that help move them around, but they're nothing like your flagella. And one of the things that they have is a lot of stuff on the surface that allows you to tell them from you. This is a surface cut through the plasma membrane here and cell wall of a gram-positive bacterium. And this thick green section is the peptidoglycan cell wall. And this cell wall has biochemical traits that are nothing like yours. Also, the lipids in the surface are nothing like yours. And so you can use that to help you decide that you've got some kind of pathogen. Even worse, this is a picture of a gram-negative pathogen. These guys also are very dangerous. They have a peptidoglycan wall. They have two membranes, an inner one, the regular plasma membrane, and an outer one. And in this outer one are lipopolysaccharides that you really will just respond strongly if you run into them. Nonetheless, these are among the more deadly bacteria, and we're running out of really good antibiotics to treat them. And so paying attention to how this one differs from you is really one of the first steps in designing some ways to protect you from them. The next one is our next group that we look at is fungi, and this is a candida shot. Fungi are eukaryotic, so their ribosomes are a lot like yours. They have an organized nucleus, but they do have a cell wall on the outside. It's nothing like the cell wall around bacteria, and it's also completely different from that of plants, and it has characteristics that you can sometimes use to distinguish them and maybe even attack them. This is another view of Candida, a little bit different magnification, slightly higher. You can see that it has both hyphae and rounded cells. And this is a different fungal infection that's inside a tissue. Another type of fungal infection that we have here would be something called Cryptococcus gattii, and that one is dangerous. So fungal infections include things like athlete's foot, ringworm, toenail fungus, yeast infections. All of these things are obnoxious and annoying, but not usually lethal. There is, however, a new fungus out called Cryptococcus gattii, and this thing is really bad. It can actually grow in your lungs, and you sometimes have to have surgery to take out masses of the fungus because it can grow so large before it's treated that it's almost like having a tumor. Okay, so in general, fungi are not something that you can't deal with, but again, it's like anything else. Sometimes you wind up with a particularly lethal case, especially if you're immune compromised. Now, this next category are those of unicellular eukaryotes. These are eukaryotes like we are. They have nuclei and ribosomes that resemble ours. They have a plasma membrane, and they don't have a cell wall. So that is something that makes these guys sometimes quite difficult to treat. They include things like Giardia, Trichomonas, various trypanosome diseases, including Chagas and sleeping sickness, Leishmania, and all of these guys are really not closely related to each other. These guys are all very different, and for each one of them you have to develop a specific strategy for that general type of pathogen. And again, the strategy development is quite difficult because you want something that kills the pathogen and not the patient. And these are similar enough to your own physiology that finding the thin end of a wedge to get in between them can be quite difficult. One of the worst versions is malaria. This is kind of a drawing of malaria showing how malaria will grow inside of red blood cells, swell, and eventually break them. And it's rather kind of interesting because the red blood cells are among your smaller cells, and yet they can grow 15 to 20 of these malarial parasites before they burst open. So malaria has a number of strategies to keep you from killing it, and we'll look at how we try to get back at them later on. Our last category is that of the multicellular worm. Worm parasites are, let us face it, icky. These are two Schistoma cercarii, that is larval forms. They're among the few parasites or pathogens that can penetrate intact skin. And these things look relatively harmless, but they're not. They can get in, and again, they will grow, take up residence in your bladder or rectum, and live for 20 years, making little copies of themselves. Oh, gross. This is a picture of Trichinella. This is the one of the infections you get from eating undercooked pork. And what you've got here are cysts of these worms that are stuck in muscle tissue. So they're encysted in the tissue where they've got a nice little hard covering around them that makes them very hard for your immune system to get to. And finally, and I'm not sure if you can get a good look at the mouth on this thing, this is the attachment site of a hookworm. And these things, of course, will sometimes attach to the intestinal wall. They'll occasionally even travel to your lungs. They're really nasty. In general, these guys like to live for years and years and years and years in you, so that they tend to do chronic infections, and you need ways of recognizing them to keep them down.
So this is my words of advice. And my words of advice are designed to keep you from getting sick in the first place, because the best defense is a defense that you don't have to raise at all. One of the biggest sources of transmission of viruses, and also many bacteria, is on people's hands. Viruses in particular are easily transferred from one surface to the tips of your fingers to your eyes and your nose. And so I would like you to get in the habit of not touching your eyes and nose with the plantar parts of your hand. That is, if you have to rub an itch on your eye, use your knuckle and make sure that you wash your hands as frequently as you can. But you're not going to be able to keep your hands clean all the time. You're going to be touching doorknobs, computer keyboards that somebody else used, handrails, elevator buttons. Elevator buttons are really a big source of transfer of diseases, and was one of the major sources of SARS transmission during the SARS outbreak about 10 years ago. There's a new outbreak of a SARS-related virus that's taking place in the Mideast. Hasn't hit the United States yet. Wouldn't you like not to be the first person to come down with it? Another thing that I should remind you of is that one of the biggest sources of contamination in hospitals are the doctors themselves. Somehow or another, it's very hard to get a doctor to wash his hands. Now, here is my hero. This is a portrait of Ignaz Semmelweis. And Ignaz Semmelweis was the man who, in the mid-1800s, told his fellow doctors that they needed to wash and decontaminate their hands very carefully before they touched women in childbirth. The doctors were going from autopsies directly into the birthing wards, and they were carrying a variety of septic bacteria, and the mortality rate where doctors were caring for people was much higher than where the midwives were caring for people. Now, this poor man went crazy because the doctors would not always listen to him. Things would go well for a while, and then they would lapse, and he'd wind up with septic puerperal fever on his wards, and he wound up running back and forth screaming, "You're killing these women, you're killing these women." And of course he was right, but he got locked up as being crazy. And so what I want to make sure is that you guys understand how very, very important it is to do all of the methods that they have at every hospital, whether it's washing your hands, decontaminating rooms with UV or other kinds of light, whatever it is, the best infection is the infection you don't get and don't have to defend yourself from. Now, washing your hands in soap and water is probably better than using alcohol gel if you've got it, but alcohol gel is not a bad thing. And the other thing I'd like to remind you is that one of the most contaminated articles of clothing that doctors wear are their ties. The white coats get washed, the shirts get washed, but the ties, they just don't get cleaned very often. My family practice doctor used to use a bow tie, and bow ties, because they don't dangle in things, are much less likely to get contaminated. They're sometimes considered the sign of an eccentric human being, but I think all of you doctors should learn to be eccentric in a way that protects your patients. I'd like you to think before you have sex. I may be spitting into the wind on this one, but having sex with somebody you don't know, and even non-intercourse forms of sex can often spread a variety of nasty organisms. And so if you're, please protect yourself and think about what you're doing. The third one is going to be a little bit counterintuitive. If you are sick, stay in bed. Because if you stay in bed, you can't spread the disease. And if you can't spread the disease, then a disease will be forced to evolve into a milder form. So let's see how this works. There are a couple of versions of cholera. The cholera that you get in water is usually much more lethal than the forms of cholera that are spread person to person, even though they're caused by the same bacteria. Why? Because if the cholera is in the water, nobody has to be healthy to spread it around. All you have to do is get sewage contamination. Whereas if you have a good program of sewage disposal and clean water, the only way to spread cholera is person to person, and that person has to be well enough to move around. Thus, the cholera has to evolve into a form that is benign enough not to kill you, or at least not to kill you quickly. One of the people who was studying the phenomenon of evolving pathogens pointed out that cold and flu medication should be labeled development of more sinister forms of pathogen medication. So in other words, if you take flu medication or cold medication and feel well enough to go into work, then you can go into work with a more virulent form of the flu than if you didn't have that medication to get you back on your feet. So if you're feeling bad and you stay in bed until you're feeling good, the chances are your virus may be forced to evolve into something that doesn't make you feel too terrible or doesn't make you feel too terrible for too long. And so, again, what you do personally matters. It matters to public health, it matters to the health of your patients, and it actually matters to the course and evolution of disease organisms worldwide.
Hello and welcome to Python for Everybody. Today we're going to install Python on a Windows system and install a simple text editor and run a simple program. We're going to go to python.org and we're going to go into downloads and we're going to download Python for Windows. So now the download is completed, so we're going to do the installation. Now we need to be in the administrator account on this Windows to do this. So we just finished the install, and so I'm going to test to make sure that we've got her done. So I'm going to go into the command prompt and check to see if Python's installed. I'm going to say py -- version, because Python would hook us back up to that app store. So now we have Python. If we type py, we'll go to the interpreter, print hello world. And then quit, open paren, close paren. Okay, so we have Python installed, so that's good. So we can close that. Now we're going to go on to our text editors. We have a number of different editors. You can use any editor that you like. You shouldn't use Word. You shouldn't use Notepad. So we recommend the brackets. If you have something like VS Code installed and it's working for you, great, keep on going. So there's other ones that you can use, but we're going to focus on brackets. So brackets is simple. It's open source. It runs on Mac, Windows, and PC. So we're just going to download brackets here. Let's go ahead and install brackets. We'll just take the typical. So we're done with that. So let's go ahead and finish. So let's just open brackets up. Well, let's go ahead and hide this. So there's brackets. So I'll just close all this stuff. Close that. So I'm going to create a file. File, new. And I'm going to make this a Python file. Print hello world, of course, the first program. And now I'm going to save the file. File, save. Now I'm going to go onto my desktop, and I'm going to create a new folder. I'm going to call this folder py4e. You'll notice it also shows up on my Visual Desktop as py4e. And then I'm going to go into that folder and name my file hello.py. And now that I've called it hello.py with that suffix, it actually does syntax highlighting. So that's kind of nice. So if I go and I take a look at this folder, I will see that there's a file called hello.py. And if I'm here, in the command, you're in a working directory. So I'm on the py4e user. So I'm in the working directory users py4e. If you're ever wondering where you're at and it's not showing the prompt, you can type cd all by itself. If I do a dir here, you'll see that I've got a number of files, including a folder called desktop. So I did cd des tab to get to desktop, and then I do a dir here, and you see that I've got one folder named py4e. Yeah, okay, and my account name is py4e, and my folder is py4e, so sorry if you're confused by that. So I can do a dir inside the py4e, but you see I'm in the account py4e, it's desktop, and then the folder py4e, and I got a file hello.py. So I can type here py hello.py to run Python. and it will say, hello world. And so that's pretty much just kind of getting started. Like I said, you can use any editor that you like, and we're just recommending that you start with brackets. So there we go. I hope that you found this a quick and useful instruction on how to install Python, how to install a simple editor in the form of brackets, and then how to run your first Python program.
To wrap up this lecture, I'd like to add a bit more context and review into the cooperation between the innate and the adaptive immune systems. Both systems pass information on to each other. The innate system evolved before the adaptive, predates it therefore, and the adaptive system basically got piggybacked on top of it in some ancient fish. However, by today, these two systems are very closely integrated, and they have evolved a complex interdependency. So, while you often see innate and adaptive systems being described as being distinct systems, one thing you need to remember is that there is a constant crosstalk between every aspect of the immune system. Here's a quick introduction to some of the interactions central to your effective response to all those microbes that view you as food. The cell labeled APC is an antigen-presenting cell, and it could be any one of several types of innate white blood cells. It's presenting antigen from a pathogen to a T helper cell, and it presents the antigen on a surface protein called MHC2. MHC2 interacts with CD4 on the TH cell. You may have read that HIV attacks CD4 plus blood cells. These are the T helper cells. The reason that this critically damages your immune response is that the interaction between an innate and an adaptive blood cell is a basic trigger in calling your immune system into action. This results in the T helper cell activating, and that means it begins to divide and form multiple identical copies of itself, a process called clonal expansion. Thus, you will wind up with a large population of identical cloned cells. The TH cell progeny spread out and alert many other cells. In particular, they will alert the B cells. The B cells will also undergo clonal expansion and differentiation and begin to produce antibodies. The antibodies may directly interfere with the functioning of a pathogen, but they also, when they bind to it, will summon innate cells that will help phagocytize the offenders and get rid of them. The TH cells also stimulate a different T cell, the TC cell. This cell has a different co-receptor, CD8, which allows it to attach to viral infected cells or cancer cells. Stimulation from the TH cell turns the TC cell into a killer, hence the term cytotoxic T cell or CTL. These cells can now kill your own cells if they are harboring threats to your health. When that happens, more innate phagocytes will clean up the resulting debris. Finally, the T helper cell will also stimulate many of the same cells that gave it information in the first place. In this case, they're stimulating a macrophage, and the macrophage will in turn send out signals to all the cells around it. These cells also will activate, and the macrophage itself will come back and stimulate the T helper cell. This produces more upregulation of the T helper cell and helps it maintain its central role in managing your overall infection. So while this lecture series will ultimately deal with these interactions in detail one by one, please keep in mind the larger context, the constant exchange of information in all parts of the systems all the time.
So one last tidbit before I sign off. And that is the distinction that I made between adaptive and innate immunity was a little naive. That is, when we look at vertebrates compared to all other eukaryotic organisms, vertebrates are the only ones that change their DNA to make different types of antibodies, and we'll see T-cell receptors. And so I use the distinction between change your DNA and don't change your DNA to distinguish between adaptive and innate. However, bacteria are capable of changing their DNA using a completely different mechanism involving an enzyme called CRISPR. What bacteria do is they actually copy the DNA of pathogens, use it to make RNA, and then use that RNA to turn off the pathogens. And it turns out that we can use this DNA for our own nefarious purposes, and it turns out to be a very, very useful tool that we now have in our kit of molecular biology. On the other hand, it's not remotely what we're doing here when we talk about adaptive immunity. So just as just for fun, I've given you the references for this bacterial system at the end of the outline. And we're going to stop here, and in the next lecture, we're going to develop the terminology that we use to describe the cells and systems of the immune system, and then start talking about the specifics of innate and adaptive responses in the next series of lectures.